Okay, well, this is really very good news. I'm Madeline Kay. I'm one of the members of Town Council, and I'm the chair of both the environmental and the resiliency committees. Um, so for all the people who are here who are members of those committees, yay. We're here today because our residents have expressed a very strong desire to learn more about our environmental and our wildlife issues. They're deeply concerned about the health of our turtles, our dolphins, our shorebirds, our bobcats. They're concerned about the threats to the beach and to the marsh, and they need to learn more about it. They know that we've got sea level rise, we've got increasing storm severity, and they have expressed via a bunch of surveys from the town and from Kika that they just need to know more about it so they can become more involved in seeking out solutions. There's a general awareness that the more that we can build our understanding and our appreciation of our unique environment, we really increase the chances for future generations to enjoy the same benefits that we all have. So that's why Kiawah Conversations has been formed. You are here today as part of our inauguration of this series of programs. And we know that our environmental and wildlife issues extend far beyond Kiawah's borders. So that's why we've invited our friends and our neighbors from Seabrook and from Johns Island. And over time, we hope to build that audience attendance. So keep an eye out for future programs that we're working on that will be on everything from dolphins and strand feeding to recycling, to composting, to alligators, to beach health. And a week from now, if you haven't signed up, there are still spots for a program that our own town biologist Aaron Givens is giving on our birding population. So we hope to see you there. And again, welcome today. I'm just thrilled at the attendance. I really appreciate you all coming. And now I'm going to turn it over to Lynn Sager. Lynn is the person who coordinated this program. She is the fearless leader of our Turtle Patrol, which is one of the oldest, most established, well-respected turtle patrols on the East Coast. And she is going to introduce our speaker today. Lynn. Oh.
Thank you, Madeline. Madeline is right. This is such an awesome crowd. I am so pleased that you're all here and that you were able to continue coming. A few folks who were ill today, and they unfortunately couldn't make it because they didn't want to be here and infect anyone else. But I'm so glad that so many of you stayed healthy and flu free. If you don't mind, I am going to read my introduction for our speaker because he is just so accomplished and so knowledgeable that I don't want to screw it up. But Jim Jordan, thank you very much for the suggestion at one of our earlier committee meetings for having Brian come Jim and speak to us. Jim noticed that the last several years, Brian has not been at our end of the year Turtle Patrol dinners. And the reason for that is because a lot of the patrollers felt the evening went on way too long with all of the presentations. So Turtle Patrollers are delighted to finally get some information from Brian because we all love the genetic information. So anyway, our speaker, Dr. Brian Shamblin, is an associate research scientist from the Warnell School of Forestry and Natural Resources at the University of Georgia. He received his MS and PhD degrees from UGA, studying sea turtle genetics, and spent a year as a National Research Council Fellow with the National Marine Fisheries Service Sea Turtle Genetics Program at the Southwest Fisheries Science Center in San Diego, California, before returning to the UGA facility. Using an approach similar to human forensics, his team has been collaborating with sea turtle patrols to genetically tag nesting loggerhead turtles north of Florida since 2006. Through nest samples, They have identified over 16,000 nesting females via genetic information from a single egg from each nest. Recently, Brian has developed a novel technique to identify breeding males via sperm trapped in the egg yolk membrane, allowing a non-invasive population count of adult male sea turtles via land. Brian will discuss how the genetic tagging has informed loggerhead turtle recovery and continues to address questions of conservation concerns with a focus on South Carolina and Kiawah Island turtles. Without further ado, Brian, thank you.
So thanks for that introduction. It's a pleasure to be here. I'm a little intimidated. I'm used to speaking about turtles with turtle folk, and I'm used to talking genetics with geneticists. But I understand, we probably have a lot of turtlers in the room, but we also probably have folks in the room that maybe don't have the background in sea turtles. So I'm going to try to carve a middle path. So I'll ask you to bear with me. I promise I'm not going to get too in-depth in genetics methods. But we do need to talk a little bit about the history of sea turtles, their life history, and some conservation history, I think, to set the stage for where genetics have really helped play a vital role. So to get started, you got to sort of zoom out globally. There are 9 major loggerhead nesting populations around the world. They're all of conservation concern. But we're really fortunate here in the southeastern United States that we have the healthiest, largest loggerhead nesting aggregation in the world. So that has been a lot of hard work to mitigate threats and to get a lot of hatchlings out in the water. So groups like the patrollers here have helped contribute to that, and I'll talk about some other folks who've made really important contributions as well. I could talk the whole 45 minutes about this, but I'm not going to. This looks like a very simple schematic in one sense. This took dozens of researchers around the world, decades, to put this picture together because everybody had little bits and pieces of it, but nobody had the whole thing until everybody sort of worked together. So I should have said that on that first slide. You saw a lot of logos there. Anything sea turtle related, it takes a village. So it truly does take collaboration across time and through space. And a lot of people had little bits and pieces of this life history, but it took sort of all working together to figure this out. So the nesting females are the most accessible life stage. They have to come ashore and find dry sand for their eggs to properly incubate. So we know a lot about the nesting females. They're the easiest ones, to get in contact with. But once those little hatchlings emerge from the nest and hit the water, it really was a mystery where they ended up. Where were they going? How long were they out there? Dr. Carr at the University of Florida called these the lost year initially, until we realized, whoa, it's more than a year, it's years, the lost years. And so we now know that those little turtles are basically epipelagic. They're at the surface of the water and they're out in the sargassum, out in the open ocean. And we think they're spending probably at least 10 years, their first 10 years in those sorts of habitats, before they ultimately would recruit back into our coastal waters and switch to feeding on things that live on the bottom of the ocean. So things like horseshoe crabs and whelks. So once the turtles choose where they're going to live, they sort of have 2 homes. They have where they came from, where they hatched, but they also have a foraging home range where they live outside of that nesting season. Sometimes those may be really close to one another. Often they're not. They may be hundreds of miles apart. And so once these females reach sexual maturity, they're making migrations from their foraging home back to the nesting beach to lay their eggs. So I should have also said this. I like to be conversational. So if you have questions as I'm going, please raise your hand so that I can see you. And I'm going to have a little bit of audience participation. So I'm sure we got a lot of turtles in here. Where do the females go when it's time to lay their eggs? Right, back where they hatched, right? So you'll often see the soundbite that they go back to the beach where they hatched. And that could be true for our loggerhead turtles. Maybe they're going back to a general neighborhood, maybe not exactly a beach. And they're probably going to do that every 2 to 3 years. They're capable of laying up to 8 sets of eggs. That's very unusual. More often, it's going to be 4, 5, or 6, about 115 eggs in each one of those nests. You can imagine that takes its toll on the females' bodies. And the whole time they're doing that, every 2 weeks, they're not eating. So it takes a lot of rejuvenation to get their body back in enough condition to come back and do that again. Usually that means they're skipping at least a year and often 2 years before they come back to try to nest again. And so the females we know a lot about, those juveniles. The males are a lot more mysterious, and I'll get into more detail on that front as we go. I wanted to start with a couple of pioneering women. If anybody has read Will Harlan's book Untamed, maybe you read about Carol Ruckteschell on Cumberland. She was out on Cumberland Island and in the 70s realized we have all these loggerhead turtles washing up dead on the beach. Those large juveniles and the adult females are the most important members of the population because they're the ones that have made it. They've reached adulthood. It's really important that they stay in the population as long as possible and produce as many offspring as they can. So losing those females early was a real detriment to the population. She started surveying on Cumberland Island just on her own volition, because she realized how important it was to document this. You know, it's a lot less glamorous than counting nests, right? Documenting all these dead, stinky turtles. But she laid the groundwork for what is now our National Marine Fisheries Service Sea Turtle Stranding and Salvage Network, a national network of folks who are out there looking for these turtles. Not too long after that, Sally Murphy, who was the South Carolina DNR Sea Turtle Coordinator, we have Charlotte Hope in the audience. I think she was in the mix pretty early as well. She's seeing the same thing up here. 1980, just all these dead turtles washing up on the beach. And it became clear fairly quickly that they were drowning in the trawl fishery. So when you're shrimping, you're dragging these big nets along the bottom to catch the shrimp. Sea turtles can hold their breath for a really long time, but also some of these drag times are really long. You can imagine if you're trapped in the net, you're probably panicking and you're depleting your oxygen really quickly. So unfortunately, a lot of these turtles were drowning in these trawl nets. This is a really good story of working with the fishers instead of having it be adversarial, but it certainly was adversarial in the beginning. I know Sally got a lot of death threats from the shrimping community. But this is Sinky Boone. He was actually a shrimper in Darien, Georgia, and he essentially invented and modified this Turtle Excluder Device. So those two big round things that you see in the nets there are a metal grate that allows the turtle to hit that grate, and they can push their way out of the net. And I think having Sinky's involvement was instrumental because he was inside the shrimping community. He wasn't coming from outside. He was a really good advocate for working with Georgia DNR and with NOAA, National Marine Fisheries Service, to get broader implementation of these TEDs. And this has made a huge difference. It hasn't completely eliminated the turtles that we lose this way, but it's drastically reduced. When these TEDs are used properly, it really helped alleviate a lot of these large juvenile and adult turtles that we were losing to the trawl fishery. And this is probably a big part of why we are seeing recovery now in our loggerhead turtle population here, but also Sinky worked with a lot of fishermen from around the world. He was training folks from Mexico and Costa Rica and Asia as well. So these turtle excluder devices have been really important for loggerhead turtles and for species like Kemp's ridleys and olive ridleys in reducing these fisheries bycatch issues. So we can sort of check the box on making a lot of progress locally. When you think about the open ocean, it's a little bit of the Wild West. So these little turtles are out in the Sargasso Sea. They're maybe making a full circuit of the North Atlantic gyre. Some of them are ending up off the coast of Africa before they come back. And there are a lot of fisheries operating there as well, these longline fisheries. These are setting millions of hooks, and they're baited with squid, which is something the turtles would go after naturally. And so it's hard to quantify how many turtles are ending up hooked this way, but we estimate that we're talking like over 100,000 probably annually. in the Atlantic. So they're not necessarily fatal, but you can imagine even small loggerheads might break your finger or take the end of your finger off. So, and these aren't vessels that typically have observers on them. So it's not impossible to imagine that somebody would rather kill that turtle than take it off alive and risk capturing it again. We don't really know what those numbers look like, but we are concerned about this. And of course, one of the things that happens in these habitats is the same oceanographic processes that are creating these weed lines are pushing anything that's floating into these habitats, which means if we have an oil spill, it's going to end up in these sargassum areas. And now the big thing that we're all hearing about are these, plastic, all this plastic pollution, microplastics. This is a turtle that had all of those pieces of plastic in its gut. Again, really hard to quantify this sort of thing, especially out in the open ocean. We don't really know what the long-term impacts are going to be at a population level, but I can't imagine this is a good thing, obviously. And that sort of brings me to talking about how we track sea turtle populations in general. A lot of the sea turtle species and populations nest at night. So you can imagine if a female comes up here on Kiawah, she's really happy about the spot that she picked. She comes out at high tide. She can dig a hole, drop her eggs, cover that and camouflage it, and be back in the water within an hour. So it's a little tricky to actually see the turtles in action. It's a whole lot easier to go out the next morning and just count all those nests. So around the world, this is how we typically keep track of sea turtle populations, is counting these nests. The problem is when you have data like these from Florida, these are index nesting beach survey data. So these are beaches where people are doing the same thing every day. They're covering the same amount of beach. So we can use these to say something about what the population is actually doing. You see the nest numbers are going up through the 90s, and then in the late 90s, they start going the wrong direction. So I started graduate school in 2004. In Florida, they bottomed out in 2007. For us north of Florida, it was 2004. When you're in the moment, the scary thing is you don't know what's coming. Like there's nothing predictive about this. It's just a measure of what's happening right now. So we were all kind of freaking out a little bit that this was going really rapidly in the wrong direction. What you really also need to know, one of the questions that NOAA had, National Marine Fisheries Service, is because those females can lay more than one set of eggs in a year, does fewer nests even always mean fewer females are nesting? So you could imagine if they're not getting enough to eat or the food quality just isn't that good, maybe they don't lay as many sets of eggs. So even a question as basic as that, we did not have an answer for. And the only way you can answer that sort of thing is to have individual female data. Track these individual females through time. And that traditionally meant flipper tagging. So when they come up to nest, you put these tags in the flippers, you put a pit tag, which is essentially like you would microchip your cat or your dog so that you know when you find that female again. These sorts of projects used to be really common up and down the coast, but there are very few of them left north of Florida. So we only have 4 really that are still active, 2 in North Carolina and 2 in Georgia. So you see 0 there in North and South Carolina. And they're logistically challenging, like I mentioned. So you really need to be able to cover the entire beach every hour from dusk to dawn to make sure you're not missing any of these turtles. The other thing that we learned from the projects that were active, and I think Kiawah was in the 70s, is that some of the females move around. They don't just nest on one island, they're nesting in multiple places. So I think these projects are really important because people are putting eyes on these turtles. And it's sort of a first line of defense to keep an eye out for things that might be going wrong, like emerging diseases and the body condition of these turtles. But we know we're missing a lot of information as well. So when I started grad school, one of the things I was trying to come up with is there some way we can get information from that nest to figure out who mom is so we don't actually have to interact with the nesting female? And if you're fortunate, like the folks on Cumberland here, and I think out here as well sometimes, you patrol the beach in your ATV or your truck or your bicycle or on foot. Anything north of Florida, if we call it a nest, because sometimes the females come up and they just leave. They don't like the conditions. It's not quite right. We call that a false crawl. I don't know why, because it's still a crawl. It just doesn't have eggs in there. But we dig down and we verify that they're eggs. That's just part of our normal protocol on all the beaches. And so basically what we found was we were working with Little Cumberland Island, who at that point was a tagging beach, and they were collecting little tiny pieces of skin from these nesting females. They had a raccoon come up and destroy a nest. And so they didn't get to see the mom, and they had all these depredated eggshells. And the technician, Jocelyn Coulter, froze some of those eggshells. And she said, hey, can you use the DNA? Can you get DNA out of these? Can you match this to our skin sample from our moms? And I was like, I wouldn't hold my breath, because I looked in the literature. There's very little in there, and most of it was what we call mitochondrial DNA. It's the type of DNA that's coming from mom. But I played with it for probably 3 or 4 weeks, and finally we ran everything, and sure enough, it worked, and it matched. It matched one of the skin samples that they had taken. So the light bulb went off about what we could do with this technique in terms of really opening up the coverage. You know, we don't have to be out there at night. So this is something that we could collaborate with everybody on and actually cover a large area. So if there is a broken egg, we try to use that wherever possible. If not, unfortunately, we do sacrifice 1 egg per nest. On average, these have about 110 to 115 eggs in them. And about usually 30 to 35% of those don't turn into a hatchling that makes it out of the nest. So I think that the management folks were like, look, this is a wash in terms of biology. It's nothing, but we could learn a lot from it. So we can justify this take. And we have been taking the shell. I'll tell you a little bit more about maybe we're going to start saving the whole egg in some cases later. We chop a little piece of that shell, we archive the rest, and then we go through this process where we're adding chemicals essentially to extract the mother's DNA off that shell. This is Aaliyah in the lab. It's sort of in one of the last steps of this process, the vacuum manifold. So we try to automate as much of this stuff as we can, because in some of the big years, we process 16,000 samples in a year. So and this is a good point for me to pause. Well, actually, let me tell you about what we do, and then I'll tell you how we're able to do it. So these microsatellite markers are the same kind of marker that we use in human forensics. So if you know anything, we have true crime junkies in here, and you know the CODIS system, the FBI's CODIS system. It's exactly the same idea, except these markers were isolated from loggerhead turtles. So we're not going to copy human DNA with these primers that we're using. We're only going to get sea turtle DNA. And this is just a fingerprint for 1 female. So we run 3 different reactions, a total of 16 of these markers. And we have enough power with this to distinguish individuals. So essentially, we're building a unique genetic tag for each individual in the population. And just to give you a little more clear example, these are 2 Florida eggs, one on top and one on the bottom. And for this particular panel, if you look at marker 73 in green, both of the peaks that occur here perfectly match. If you follow the one from the top down underneath, you see the same 2 peaks there. So we have a match. And so when we have that sort of thing, we want to keep looking at additional markers. If we look over on the other side at marker 28, you notice that the front peak matches, but on the back end, we have 2 different peaks happening. So we don't have a match here. So we can use these markers to resolve different individuals, but also these markers give us some basic information about how related the females are. And in this particular case, the pattern of matching that these 2 females had was consistent with what we would expect from sisters. So these 2 are sisters. They have the same mom, same dad, but different females. Yes? That's a great question. So one of the reasons that we do it is because if we don't have a sample from that nest on the front end, we may never have an opportunity to get a sample. A lot of these nests get washed out and we can't find them, or they get completely taken out. The other problem is that if you got DNA out of the nest at the end, you're getting a mixture of mom and hatchlings from those eggshells, if you get anything. What we tried to do initially was use any eggs that didn't show any sign of development, because those should just be mom. But the worst case scenario for DNA degradation is hot and wet. And guess what all these sea turtle nests are? They're very humid. It's really, the only thing worse would be if they were out in the sunlight. So sometimes it works, but I would say more often than not, it fails. We really tried hard initially not to do that. The other thing that we tried to do was just swab the fluid off the outside of the egg. Now remember, this was 20-something years ago. I think if we revisited this now, we might have better success, but with everything that we had at our disposal 20 years ago, that was long before we were talking about touch DNA with things like human forensics. So again, we didn't love it, but everybody felt like it was worth it to get the information that we're getting. It would be interesting. We are actually working with diamondback terrapins as a surrogate to try, because they don't lay very many eggs. We don't want to sacrifice those eggs. We're trying to use those to see if we can get enough DNA off the outside of the egg to identify mom. So here's where I wanted to pause. I'm going to tell you about data from essentially 1,000 kilometers of coastline, from marine land in Florida all the way up through North Carolina. There is no way we could do a project like this without tremendous buy-in from the state agencies and without everybody that's out on the beach every morning counting all these nests. So for those of you that are patrollers from out here, thank you so much. Everything I'm going to tell you about would not be possible without your contributions. And the same thing goes for in the lab. Given the volume of samples that we work with, there is no way that, I'm not a one-man show. I believe we've hit 90 different undergraduate students now that have worked on this project since the beginning, and a lot of great lab managers, technicians over the years. So this really is a huge village that's contributing to these data. I don't want to dwell on this too much, but some of my graduate work was trying to resolve population boundaries. Now, sea turtles don't read these papers, so unfortunately, they don't always do what we think they're going to do. But the different colors that you see here essentially are different female families that we're tracking. So on one hand, we don't have a lot of diversity. You basically see a lot of blue and yellow here. But if you look at the relative frequencies of those as you go southward, you see that all of our turtles up here north of Florida are the type in blue. And by the time you get all the way down into the Bahamas, you see none of those. They've completely switched over to the turtles in yellow. So we can use these sort of data to make an inference to say, hey, if you hatch down in central Florida, down where the CAN is, Canaveral, you're probably going to come back somewhere in that general area when you start nesting. And we feel the same about most of our turtles north of Florida. They may not come back to a very specific beach, but they're coming back to a general area that's probably a handful of islands. And so everything I'm really going to talk about today is the Northern Recovery Unit. So it's by the book, it's all those turtles north of Florida. Again, they don't read the books. We think some of those turtles are also nesting down on Amelia Island and down through Duval and St. Johns County in Florida as well. That's what this looks like. So we started statewide in Georgia in 2008. We added South Carolina and North Carolina in 2010, so we've been doing this for a while. And it took a little while to get permission, but we were seeing some interesting things in North Carolina that made us think we were missing some of those turtles. And it turns out they're nesting in Florida. So they're sort of bouncing between Florida and North Carolina. They're kind of skipping over us in Georgia and South Carolina. I'm not going to talk about that today because we don't have time, but this gives you an idea of the scale of the coverage for this project. And again, there is no way you could do this at night, and there's no way we could do it without all the volunteers that are out on the beach every morning. So to date, we have assigned 129,000 nests to almost 15,000 unique females in this population. And if we zoom in just to Kiawah, that's nearly 5,000 nests and 1,000 moms. Different moms have used Kiawah since the start of the project in 2010. And to circle back to that question that Noah started out with, do fewer nests mean fewer females? The answer is yes. We figured out very quickly that there is a really strong correlation between that nest count and female count. And so it is a pretty good way to track what's happening right now. Can't really predict what's going to happen in the future, but it's a pretty good translation. So if we go back to that record low nesting year in 2004, the number in the sand-colored track is the number of nests, and that darker blue underneath it is the number of unique individual females each year. you see that the numbers in general look really good. We haven't seen another year like 2004 since, but there's also a lot of variation, right? So we have these high years and low years. If you notice, we tend to have peaks on 3-year cycles. So if you look here, 2013 was a big year, 2016 was a big year, 2019 was a big year, 2022 was a big year. So we were ready for 2025 is just going to blow the roof off. And then it's like, well, again, the turtles did not read what they were supposed to do. So we're not panicking about this yet. Again, sea turtles, anything that you do with sea turtles, you better be in it for the long haul. We're not really sure exactly why this happened, but I can show you some of the background I think that's contributing to this. So if we look at females that nested in 2025, 5% of those were new. They had not nested prior. This number is going to go up. Probably we haven't totally finished all the assignments yet. And they did what we expected in the sense that 66% of those females that nested last year were last here in 2022, 3 years prior. But if you go back to 2022 and you ask, well, how many of those 2022 females nested in 25? It's just barely over a quarter of them. And so then you ask, well, what about 2019? And if we go back to 2019, half of those 2019 females were back 3 years later in 2022. So we just didn't see that cohort that we expected to come back. Are they still out there? Most likely. I don't know why the delay, but again, it takes a lot of energy to yolk 1,000 eggs. So you can imagine if conditions were just a little off, it may take them an extra year. So I fully expect they are going to come back in full force in 2026. But again, we'll see if they make liars out of us or not. The interesting thing about the genetics to me, in a lot of ways, the data that we're collecting are just like if we were tagging these turtles with physical tags. But the relatedness bit is where we get that extra information that we would never have gotten from physical tagging. And what we're seeing is new females, the proportion of new females that we're seeing every year is going down. That's not surprising. There is a carrying capacity out there. There's only so many turtles that are going to be able to live. And so the longer we do this, the fewer new females we're seeing every year. But the ones that we are seeing, an increasing proportion of those we actually can assign to a known mother in the database. So in other words, we have new moms coming in. It's the first time we've ever detected them with the genetics, but we're able to match more and more of those to existing moms in the database. And to me, this is really the value in this project. We're going to be able to start answering things that have been very tricky to get to. And one of those is that idea of natal homing. So what do these females consider as home? And that seems to vary across species and in different populations. So here's another bit of audience participation. I'm sure you've heard sound bites about X percentage of these hatchlings are going to reach sexual maturity, right? So throw some numbers at me.
What?
1 in 100, 1 in 1,000, 1 in 3,000, 1 in 5,000, 1 in 10,000. Yeah, it's tricky, right? The reason there's so much variation in that number is how do you get at that? There is no easy way really to get there. So the interesting thing that we found, this group of new females from 2016 to 2021, one of the things I did was just ask how related are they? How many close relatives are in this group of 5,000 females? And what we found is 20% of that 5,000 had at least one full sister in this recruitment cohort. It gives us a little bit of context. So the question is, if we are able to do this matching, can we tell how old these turtles are? The short answer is no, right now. There's a lot of work being done on epigenetics. So the epigenetics are not the DNA sequence itself, it's all the switches that are turning those genes on and off. And so there's been a lot of interest in epigenetics and cancer research and human aging. And so there's been some work done with green turtles in Australia where they are trying to identify markers in that epigenome that we could use to actually age turtles. And that would be awesome. I know that NOAA, National Marine Fisheries Service, is working on that right now. So unfortunately, no. Unless we have some tagging data or something that we can sort of corroborate with, we don't know. And if we only have a pair, if we only have one turtle that looks like a mom and daughter, a pair of those, then we don't know who's who, right? Just because we got one earlier doesn't necessarily mean that's mom, because they can stay out for years before we see them again. We had a few females that nested in 2025 that we had not detected since 2008 when we started the project in Georgia. So we can't write them off either. You would think if they're not back in 3 years that, well, they're done. They've died. But they're doing all sorts of weird things that we didn't expect. And so, unfortunately, it just gets, the mysteries get deeper with more data in some cases. Absolutely. So the question is, could they be nesting somewhere else? Again, the dogma is they go back where they hatch. And that was the first thing that we noticed in North Carolina was as you get up into Cape Lookout and Cape Hatteras, we're seeing those females lay fewer sets of eggs. We're seeing them lay them over larger areas, and they're much less likely to return. A lot of those turtles we see once and we never see them again. So we went through a whole list of possible reasons for that. And way down on that list was, well, they're nesting somewhere else. But they don't do that. So we kind of wrote that off initially. Well, now we know they are doing that. And the somewhere else is the only place we're not looking, and that's Florida. So Central Florida even. So there's one documented tagging record of a turtle that was tagged at Cape Lookout in North Carolina, and 21 days later, Doc Earhart's crew got her nesting at Cape Canaveral. And we have detected turtles actually bouncing back and forth between North Carolina and Florida. They just skip right over the top of us. So we're watching the loggerheads and green turtles actively colonize the northern part of the North Carolina coast right now. And that's not something that we would have predicted when we started all this work. So I can't show you all of these turtles. I wish I could, but I did pick one representative family that I think is really cool with the relatedness data. So this is a mother that last nested on Kiawah in 2020. And these are 4 of her daughters that are recruited into the nesting population. So, but there's 5 because this female has actually mated with 2 different males to produce these new females. And in turn, that second male that she mated with is mated with an unknown female, and we have one of their offspring out there nesting. And it gets really interesting when you see where these turtles are on the map. And the reason I picked this family is we're seeing a lot of these sorts of large families in the data. So, of course, one of the questions is there something magical about this mother's genetics that are allowing all these daughters to make it? I would argue that it's probably more environmental. It's about that incubation environment. So it may be something special about where mom is putting those eggs, more so than anything about her genetic makeup. The other thing interesting here, notice that all of these daughters recruited between 2019 and 2021. So we do have some moms that were tagged way back in the 80s that died a long time ago, we think, that have daughters that sort of recruited in 2015 and 2016. So we get this pulse like this of 2 or 3 sisters, and then we see nothing else. So it could really be that a lot of our moms, this will be it. I mean, she's done her job, right? She's replaced herself 5 times over. That's fantastic. That's a growing population. But it could be that a lot of these other mothers are nesting for 20, 25, 30 years. We will never see an offspring of theirs hit the beach. And again, we don't know why. But these are the kinds of things that we can learn with these long-term data with the genetic markers. So let me pull out and show you where these turtles are on the map. So this is mom, and this turtle did not have strong sight fidelity. I could have shown you lots of turtles that every single nest we've ever recorded for them are right here on Kiawah. She actually nested down on Ossabaw Island in Georgia in 2009. She may have nested up here. We weren't doing anything in South Carolina quite then. And then in 2020, she laid her last couple of recorded clutches on the north end of Kiawah. So 2 of her daughters are nesting to the south. And interestingly, both of those daughters at some point nested on Ossabaw Island. So she probably was doing that long before we started that genetics project in 2008. Even when we find females that are using multiple beaches, they tend to do it very consistently. This is not a mistake. They know what they're doing. We don't know why they're doing it or how they're doing it, but they know what they're doing. And it's probably bet hedging. You know, don't put all of your eggs in one basket, right? And then the other daughter, we have one that's right here on the south end of Kiawah and over on the north end of Edisto. Two more that are on, one to the north, and then another that is using Kiawah but also has nested even north of Cape Romaine. So you see here that for any one of these turtles, they're not necessarily using one single beach. They're using multiple beaches. And you can see, even though we think all of these females came from the same mother, they really have spread out over a large piece of the coast. The other daughter that I did not put on here, of that second male, is nesting on Ossabaw, right where this turtle in brown is as well. So we really do think that mating that happened was happening right offshore, and I'll show you more of that momentarily here. So the males have been the big mystery. So the females have to come on land to lay those eggs. That gives us an opportunity to learn more about them. The males, if they're healthy, we're never going to see them. You know, they're out doing their thing in the open ocean or on our coast. So while I was in California for that postdoc with NOAA, two of my office mates were Cameron Allen and Michael Jensen, who had worked a lot in Australia. And this is a paper that they published. The largest green turtle population in the world nests on Rain Island in the Great Barrier Reef. All those little specks that you see there are nesting green turtles. So it's incredible, the nesting density that's there. They were able to team up using genetics and some endocrine data to show that no males have hatched on that beach for at least the last 20 years. So sea turtles have environmental sex determination, hot chicks, cool dudes. The hotter, drier nests are producing females. The cooler, wetter nests can produce some males. And so this is such a tropical beach. Not only is it not producing males, it's also cooking, unfortunately, a lot of the eggs. Like the nests are getting too hot. And there's been at least some concern about this in loggerheads here in the southeastern U.S. So these are some data from Jeanette Weineken's lab. She's done a lot of pioneering work where they raise the turtles in captivity until they're large enough that you can actually sex them with laparoscopic work. And there are some years where their sampled nests are producing only female hatchlings. So the other thing about this is not only are they all female, a lot of these really hot, dry nests are producing smaller turtles. They can't right themselves as well. They can't crawl as fast. So there's part of why there's so much pushback from the managers about, well, don't move all these nests. Don't relocate all these clutches. So it's a conservation measure. If a female lays her eggs too close to the water, we always wanted to move that higher to make it more protected in the past. Well, now it's like, well, maybe we need to leave some of those that are marginal where they are, because these females sort of know what they're doing. They've been around millions of years. Maybe if that nest gets washed over a time or two, it'll cool it off and we'll get some male production. So there's been a pushback about management, about moving all these nests, because we don't really know what we're doing to that environment. And for years, we were worried about getting hatchlings out. We got to get the numbers up, get a bunch of hatchlings in the water. Well, now we're learning it's not just about numbers, it's probably about the quality of those hatchlings. Because if you're small and you don't swim very fast, you are basically the Cracker Jacks of the sea, right? All these fish are going to really eat you up. The other thing that we've learned from Dr. Weineken's work, where she's kept a lot of these turtles in captivity for a while, not only are they smaller, but they grow more slowly. So it really makes them vulnerable to being eaten by a lot of predators. I say all that to say we don't really know much about the males. We don't know how many males are out there breeding right now. We don't know what percentage of our hatchlings leaving the beach are male. But obviously this is something that we would like to learn more about. The only data that we have on males is coming from hatchling sampling, where we use genetics to figure out how many dads were in the mix. This is some work by Dr. Jake LaSala, who leads Moat's program down in Florida now, but this was when he was a master's student at Georgia Southern. He worked with Coretta Research Project on Wausau Island to sample a lot of hatchlings. He found that 75% of the nest had more than one dad involved, and on average, it was 2.6 males per female. Over the course of 3 years, they identified 195 different males. They never found repeats across females or across those 3 years. So what does that mean? You know, what do you take away from that? Do you say, well, 2.6 males per female, can we just multiply that by all the females we've identified? That would be one way to do it. What that ignores, though, is whether those males are also mating with other females. So the multiple paternity thing is called polyandry, where you have a female that has mated with multiple males. If you're sampling several hatchlings from a nest, that's very easy to detect. You have a really good power, right? You're gonna pick that up. The problem is the flip side of that, what we call polygyny, which is, yeah, those same males are probably out there trying to mate with all the females, not just the one that you're looking in her nest for. And so no polygyny has ever been documented in the southeastern US, but it has in Brazilian loggerheads and some other small populations of things like hawksbills in Asia. So we started a pilot study in 2024 where I'm sorry, I know I'm greedy and I ask a lot of y'all. I said, we don't just want the egg, can we save tissue from dead hatchlings? When you go out, after the hatchlings have emerged from the nest, we go out 3 days later, we dig everything up and we try to count how many hatchlings do we think made it out of the nest. And in doing that, we can salvage a little piece of tissue from those dead hatchlings and try to, since we know who mom is, that tells us a lot about the potential dads. The other thing is this whole question of where exactly are they mating and are there opportunities for them to exchange genes over large areas sometimes. So many of you are also aware that Kiawah participated in this stable isotope study. So stable isotopes are, you are what you eat. We can use yolk from that one egg to tell us which area mom is actually living in when she's not at the nesting beach. On a big scale, at least. So if you look at this map here, you see 4 colors that stand out. And Kiawah, along with all the other South Carolina data, most of these moms that are nesting here go south to that area up in purple, the Mid-Atlantic Bight. This is a really productive area. We do have a little bit of data on males. This is some work by South Carolina DNR's in-water program. All these males were trying to breed down at Cape Canaveral in Florida. The numbers that you see on the map are where they live outside of breeding season. So if you compare that to what the females are doing over here where it says CE, you see there's a lot of overlap. A lot of these males are actually going to that same area where our females live. So you can imagine, the other thing that we learned from this telemetry data from the males is the males are leaving their home range and they're setting up shop at the beach before the females get there, and they're just waiting on the ladies to come by. So there's mating happening between those males and the females that have settled out, because the females are getting to the beach about usually a month before they start laying eggs. But then there's also opportunities for those males to catch females on their way north and females on their way south. So we have the potential for mixing to happen over very large areas. And these are some data from that 2024 pilot study. So these are just salvaging tissue from the dead hatchlings. Every line that you see here is where we have a shared male that was assigned as the father of hatchlings on those beaches with different moms. So Cape Romaine was the hub. So Lighthouse Island, Cape Island, there were 15 males there that mated with at least 2 different females that were laying their eggs there on those islands. And we saw connections even between Jekyll Island, Georgia, as far north as Bald Head Island, North Carolina. We had hatchlings coming off the beach that had the same dad. So in this case, what we saw was 565 hatchlings from 324 different nests, 245 moms, 241 dads. So it's something much closer to a 1-to-1 sex ratio, not those 2.6 males per female. And that may be fine, but part of the argument that a lot of folks have made about multiple paternity is there's plenty of males out there and the males are breeding more often than the females are able to lay. And all of that will help mitigate anything that we need to worry about with climate change. And the reality is they're much closer to balanced. So it's not necessarily a bad thing, but it's good to know where we are now. And so we can track this moving forward to see if it changes. This is one really cool family group, and it's sort of complicated. I'll walk you through it. It'll be a lot easier to see on the map, I promise. This mom in orange to the right, all 3 of her sampled nests from Kiawah had the same dad. The dad 2 in light blue, but dad 2 also mated with 2 other females. So he mated with the female that ended up nesting on Seabrook in brown, and the female in pink that nested over on the north end of Edisto Island. That female in pink in turn mated with this dad one in dark blue, who in turn mated with the mom in green, who in turn also mated with dad three. So this is why I called this the young and the not so young and the restless, right? This is like a TV soap opera. But it really shows the importance of understanding that, yes, these males are mating with other females. There aren't as many males out there as we would think if we just narrowly looked at this from the perspective of a single beach. And here, hopefully this makes a little more sense seeing it this way on the map. So we have the Kiawah mom here in orange. All three of her nests, the hatchlings that were sampled belong to this one male, but you also see that he mated with the female that nested on Seabrook and over on Botany Island. And so when you see patterns like this, again, we don't really know where any of these turtles are, but I think it makes a lot of sense that they're probably mating right offshore here to see this clustering the way it is. But I'm really happy with these data. I'm going to keep begging for more dead stuff to keep doing this kind of thing. The tricky part, though, is even when you know who mom is, if you're only comparing sort of a dead hatchling from over here and a dead hatchling from over there, there's still some uncertainty. There's still some fuzziness about some of these assignments. And I started thinking about it. It was like, man, it would be really cool if there was a way we could directly get these males like we do the females. We could build a male database with his true fingerprint, the whole thing. So I was at a poster session at our Southeastern Regional Sea Turtle meeting, and Sean Perry, who's listed here, was a veterinarian at the Mississippi Aquarium at the time. And he said, in captivity, if we have turtles and the egg doesn't develop, we want to know if it was fertilized or not. If you're managing these endangered species in captivity kind of thing. And he's like, what they do is they take the yolk and they take the membrane around the yolk and they stain it and they look at it under a microscope to see if they can see sperm. And if they see sperm, then they know it was a fertilized egg. And he said, if they can do that and see it under a microscope, can't you genotype it like you do with the females? And I thought, hell, that's a pretty good idea. So I started trying to do it. And unfortunately, I didn't really have a lot of context. I had no hatchlings. So I wasn't sure if I was interpreting things correctly. And then I told Simona Chiriani this, who leads Florida's sea turtle nesting program, and she kind of forced me into figuring this out really quickly because they started taking eggs in Melbourne Beach, which is sort of the epicenter of loggerhead nesting in Florida. Cheryl Sanchez did that piece of this, and then we also collected a lot of live hatchling samples, so we'd be able to tell what are we actually seeing in this. So what you're looking at there, that thing that looks kind of like a contact lens, that is this thing called a paravidulin membrane. It's basically the membrane that surrounds the yolk and that keeps the yolk from breaking. It's the thing that you really get aggravated when you're trying to fry an egg and it breaks and the yolk goes everywhere. Sea turtles have that too, and a lot of those sperm are trapped in that membrane. So what we did is we tried to optimize how we were getting the DNA out of that, and then Cheryl was screening a lot of their nests when they thought they were getting ready to hatch, and then taking these tiny little skin samples from the hatchlings, and then getting them back to the water as quickly as possible. So we have data on about 1,000 green turtles and about 600 loggerheads for this. When things work really well, what you're looking at here is mom's DNA on the top from the eggshell, dad's DNA on the bottom from that yolk membrane. So sometimes we get mom mixed in. It makes it a little tricky. This was a nice clean one where if you see those two peaks for mom on the top and you follow them down, you don't see them down below. And this was an easy case to interpret that we had one dad. We only had one dad involved in this set of eggs. And so we have dead hatchlings that we salvaged, and notice that all of mom's alleles are here, all those female peaks, and then all the male peaks. So we got exactly what we expected from these offspring. It's not perfect. It doesn't work every time, but it works most of the time. The only turtle that we completely missed, not being able to detect this male even existed, was a male that had one hatchling out of 101 green turtle hatchlings. I can live with that all day long. So all these other males, even if they only contributed less than 10% of the clutch, there was enough of his DNA in that yolk membrane to tell how many males were in there. So the only thing that we can't get from this is we don't know where those alleles are coming from. So if we have more than one dad, we can't rebuild his fingerprint, but I can tell you, well, there's at least 2 dads in here, 3 dads, 4 dads. I think for the green turtles, we're up to 5 sometimes, males in one nest. So we're going to start using this. Florida already has started using this as of 2025 as sort of an index of how many males are out there. So it's something that we can really scale up. We don't need the live hatchlings. Again, we're sacrificing that one egg, but we're not interfering with mom. We're not interfering with the hatchlings at all. So we think it makes the most sense to use this to get the information that we can. One of the questions that we had, though, all of these Florida eggs were in like a 10-day window, basically. So we only had mom in there once. So one of our questions was, if you take the egg from the beginning of the season versus the end of the season, are we going to get the same answer for the dads? And so that's something that we asked on Jekyll Island this past year. I don't want to go into a ton of detail on this. I'm only telling you this because we got 23 males that we were able to directly fingerprint this way. And when I started looking at all the hatchling samples that everybody sent, we found 2 of those dads fathering South Carolina hatchlings. And in this particular case, it's a mom nesting on Edisto, the mom there in pink. She mated with 2 different Jekyll males. So again, we don't know, necessarily where any of this is happening, but I think it makes a lot more sense that she was passing by Jekyll because we're not detecting these males anywhere else up here. So you can sort of see this has the potential to be really cool for building a male database. We can get an idea of how many males are out there, and we can start to build all these spatial connections about where we think this mating is happening. These are really preliminary data. Long way to go with North Carolina and Georgia and additional South Carolina data, but I wanted to show this to say that you all have shared 7 males with Edisto. So they fathered hatchlings over on Edisto, but also on Kiawah this past year. So I think this is going to be a really powerful thing, and this is my pitch. We had Hilton Head sort of helping us try to learn more about the process this year. Based on what we've learned on Jekyll, I feel much better about the technique now, and I'm going to make my pitch that I think Kiawah needs to be an index site for some of this male work so we can kind of see what's happening. And sorry, I should have checked about the logo, but I just wanted to thank everybody. I want to thank the town for helping with Lynn's budget now. The Conservancy did 2 really big rounds of fundraising. We would not be able to do this. We don't have federal funding anymore to do any of this work. We are surviving completely on donations for this stuff. So huge thank you to everybody who donated and everybody who helped, collect samples, all the blood and mosquitoes and everything that happened out on the beach. The QR code will take you to my lab website. And I've been really terrible about updating, but I know a lot of volunteers are on Facebook, so we do have a Facebook page that I try to post updates on as well. I saw some hands.
That nested, and I was lucky enough to be there when they hatched. And the difference in their energy as compared to the loggerheads was striking. Is that because they're green turtles, or is that because our loggerheads are tired, or something else, or not big enough, or something? It was stunning. They just took off for the water, and their little heads came up, and they took off. I mean, it was like we didn't have to encourage them. And usually with the loggerheads, they're exhausted, of course, because they've been in there for so long. That's 3 days, but still, it must have been the same with the green turtle. I don't know. All I know is it was quite stunning, and I wondered if you had thought about that.
Yeah, so green turtles, the question is, differences in behavior between green turtles and loggerheads. A lot of people who work with both really hate green turtles. They're very spooky on the nesting beach. Loggerheads are pretty sedate. You can, send them back to the water if you do the wrong thing. Green turtles are always the nesting females are always sort of looking around. They don't want to be touched. I've seen in Melbourne where a green turtle was trying to nest and a loggerhead bumped her, and it's like, oh, I'm done, going back to the water. And they have the green turtles have the simultaneous front flipper gait too, so it's almost like watching a horse gallop, like you're talking about. They can pop wheelies, come off the ground with that. So that's just normal green turtle behavior. That's green turtles doing green turtle. Their nests look like somebody threw a grenade on the beach compared to a loggerhead, because they just get all the way down in the body pit. And they sling so much sand when you're trying to look from a distance. The loggerheads are easier to tell from, down at the water line. Okay, yeah, she's covering, or no, she's on the front end. But the green turtles, they're slinging so much sand everywhere. I'm like, I don't know what she's doing.
It was very exciting.
Yes, sir.
How many years does a loggerhead have? It's over what period of time, and is there a certain age which they don't reproduce anymore?
I think we think they don't senesce. So as far as we know, they're out there nesting until they die. That's true of a lot of reptiles. How old can they live to be? Like, how long are they nesting? That's a great question. I don't think we know. The longest one that I am aware of is a turtle that was tagged on the north end of Cumberland. I sampled her actually on Jekyll Island as part of my master's research. My partner Bree actually went through a lot of old data that wasn't in the database, and we realized she actually was tagged on the north end of Cumberland in 1980, which is the year I was born. And since I saw her on Jekyll, I had a skin sample from her. She nested only on Cumberland after that, and we followed her to 2016. So she nested for at least 36 years. But we don't know if 1980 was really her first nesting season. It could have been even longer. And unfortunately, we haven't seen her since 2016. But again, I told you about some of these females that have been sort of out in the ether forever. And I keep hoping every time I look at Cumberland data that she'll be in there. But so far, not so much.
I had a question for you, Brian.
Yes.
Why does the temperature determine the gender of the turtles?
I am not a physiologist on that front. They just don't have sex chromosomes. So it's, again, those epigenetics, it's certain things that are getting triggered to send them down different developmental pathways. And there's work on that as well. So that's a big problem. We want to know how many male hatchlings are leaving the nesting beach, but you can't sex them externally. There are folks working on epigenetics to answer that question too, so that maybe we could take a tiny blood sample or a tiny skin sample and tell whether it's a male or a female. Yes.
How many other labs are doing genetic research like you, and where are they located? How do you all done?
There are folks, so the question is, how many other folks are doing genetics? There are a lot of sea turtle geneticists around the world. I don't know of very many projects that are doing exactly what we do here. I think there's a couple in the Mediterranean in places like Italy. And I've heard from my colleague in Australia that some of the indigenous communities on the top end of Australia are actually doing it with some of their olive ridleys, because it's just so logistically difficult to get out there. It doesn't make a lot of sense for them to try to intercept the females directly. A lot of it tends to be more population based. So not trying to get all the individual turtle data necessarily, but understanding population boundaries. And then once you establish where the populations are nesting, where are those turtles through that whole life cycle? So we try to take samples from turtles in water, figure out where are they nesting, and then we try to, piece all of those pieces together to get like that whole life cycle and how that comes together spatially. Yes, sir.
So what is your best estimate? 1 in 100, 1 in 1,000, 1 in 10,000?
I'm not even going to hazard a guess on that. I think we're getting close to be able to crunch some numbers, though, with some of the relatedness data. I think it's really, so early on, and even still now, if we don't have daughters recruiting, it may just be because we haven't waited long enough, right? Like the family that I showed you all, mom nested in 2020. These turtles take at least 25 years to reach sexual maturity. So I'm going to have to do this for the rest of my career to know if she has any more daughters come back or not, right? That's the tricky part, and that's why those numbers all get thrown around, because it's very hard to really pin down. And then the other bit of that is we're basing all that on the females that are coming back. What about the males that are out there that we don't? But I will say, and I don't think I mentioned this, if they're full siblings, that means same mom, same dad. I think that means they hatched the same year. I think the odds that you have those two different individuals mate across different years in a population as large as ours is very low. So I don't know what year it was, but I think it's the same year. And if they can get these epigenetics markers working to age, then we have all these nice family groups where we know when they recruited, and we'll be able to maybe get those answers of exactly how old they are.
And I think one more question.
Yes, Charlotte.
So if you're saying the new nesters, was that 30% had some relatedness to a known one?
20% of the new moms have at least one sister that also recruited.
Okay, so does that mean then these recovery units are pretty close genetically, right? Closed system, the populations?
Except for, these Florida turtles that are nesting in North
Yeah.
Carolina.
It's the males, are they the ones that are sort of bringing in the different genetics, or?
I think so. So I didn't get into that, but we have this really strong structure with mitochondrial DNA where we can tell everything apart really easily. If you look at the nuclear DNA, just in terms of allele frequencies, you can't tell the difference between the Florida Panhandle and North Carolina. It's one giant melting pot. So yes, it could well be the males. And I didn't want to spill the beans yet because Florida hasn't had their permit holder meeting, but since you asked, some of the males that we picked up in Melbourne Beach have fathered hatchlings in South Carolina. So we do have those, at least, connections happening at that scale already, just no more than we've sampled in Florida so far.
Okay.
I have one more. It was just triggered. Can I ask one more question?
Sure, go ahead.
The loggerhead turtles mate and she stores the sperm inside of her. Could it be a coincidence that the male and the female are just mate and have the daughter from that one egg and one sperm, but the next eggs that she lays, could that have been fertilized by a different dad?
So that's something that we looked at on Jekyll Island with the PBMs, especially around the yolk. And again, that's going to be kind of the second paper we work on. Everything looked like mirror images. It didn't matter if it was her first nest of the season or the sixth nest of the season. All the mating's happening on the front end. Those sperm are mixing very well. And so they look, the sixth egg that we sampled, the sixth nest, looked just like the first one, the beginning of the season. Which is the best case scenario for us, right? Trying to use it as a technique means it doesn't matter when we sample that egg, we're going to get the same answer, which is great.
It's good.
Some of the people, Brian, they're not turtle patrollers, don't realize that the female is not going back into the ocean and mating again. Like you said, the mating is all up front.
There's some folks who have hypothesized that there's some remating because of the hatchling sampling. I think based on what we're learning on Jekyll, it's just sampling error, basically, because these eggs look the same to us across the whole nesting season.
So there's a mating season, and then there's a nesting season, and they're mutually exclusive.
So those satellite tagged males that the South Carolina DNR put tags on, the females are showing up to the nesting beach about a month before they actually start laying eggs. Those males, by the time the females started nesting, were back home. Yeah, the mating is in a really tight window, at least for our loggerheads, we think. And then the males are done. They're back on their foraging rate.
The question I was going to ask earlier was about sharing data across all of the loggerhead nesting spots around the whole world, and then comparing whether our turtles here are actually going somewhere else.
So the mitochondrial DNA helps us a lot with that because at a big scale, they tend to be really unique. The only place where we know that's happening with our turtles is in the Western Mediterranean. So most of the Mediterranean nesting is in the east, like from Greece eastward. Some of the rare nesting that they have in places like Spain, on the Spanish coast, inside the Mediterranean, they're our turtles. They just didn't come back home. They made it over there and they're like, I guess this is good enough. I don't really want to go all the way back over there. So they're essentially colonizing that area.
So they are traveling long distances.
Oh, absolutely. And all the Mediterranean green turtles are haplotype A13, which means nothing to you, but that's a Caribbean haplotype. It's very rare in Florida and Cuba now. I hypothesize that was a really common haplotype that could have existed in places like Bermuda, where they wiped the nesting population out due to overharvest. And so just a few females, you know, that were in there as juveniles decided not to come back home, and that's how they colonized the Mediterranean. So it's happening. We thought it was really rare, but now I'm actually watching it happen in North Carolina now from Florida with both loggerheads and green turtles. So it's good to know they're flexible, right? It's an insurance policy. Don't put all your eggs in one basket. You know, it's great to have really strong site fidelity if everything stays the same and it's perfect. But what happens when you come back home 30 years later and the beach is all destroyed? You know, your population would go extinct very quickly if you didn't have at least some females that were moving to different places.
Do you think changing water temperature has anything to do with that? I mean, moving north from Florida to North Carolina, I wonder.
So we always thought the northernmost areas were the youngest because of that. You know, during periods of glaciation, it would have been too cold, even here, for the turtles to incubate properly. So, but the vision that I think all of us had in our head was they're sort of slowly working their way up the coast, and instead what we're seeing is this thing where they're going from Cape Canaveral all the way up to Cape Lookout and Cape Hatteras. And to me, that's because of the shape of the coast. They're going cape to cape, where it's easy. I'm not going in that Georgia Bight and South Atlantic Bight. I'm just going to, you know, hit the parts that are sticking out. I don't blame them for, you know, they're doing a lot of work. So if that's what they want to do, great.
Thank you very, very much. There are several people that comprise the village that makes this kind of program happen. If you are a Turtle Patrol member, if you would stand up, please.